Welcome back. Here's another topic review video for you. We are now on Unit 7, the first genetics unit. And the first prompt of this unit asks us to explain the function of nucleic acids. And when we're talking in terms of genetics, there are two nucleic acids that we care about. That is DNA and RNA. DNA being deoxyribose nucleic acid, RNA being ribose nucleic acid. Okay? ATP is also a nucleic acid, but it doesn't function in the same way that DNA and RNA do, and it doesn't play a role directly in genetics. So we're ignoring ATP for this unit. So DNA and RNA, their job is to store and transfer our genetic code. That's what nucleic acids do. The variety of their details combined with the uniformity of their overall structure makes them ideal for this purpose. The small change in nitrogenous base allows for these nucleic acids to effectively function as our letters in our alphabet do. Right? And so you can basically spell out instructions for making our traits the way they are supposed to be, utilizing nucleic acids. Our second prompt that we're going to look at asks us to create, analyze, and explain a model of DNA from the level of double-stranded structure to the gene level. I've modified that ask a little bit. So we have a gene sequence. Right? So this is the specific order in which our nucleotides are showing up within the DNA. And so this varies for all of us individuals. Even within the same gene, you're going to have changes in these individual sequences. Right? And now, if we have the same allele for the same gene, we'll have the same sequence. And so there's a specific allele that codes for blue eyes in that gene. And so my blue eye sequence is going to be pretty much the same as somebody else's blue eye sequence, barring a few opportunities for silent mutations and such. But overall, same sequence. Okay, so that's a gene, a region that codes for a specific trait using a specific sequence. Okay. If we zoom out a little bit and we look at the structure that we'd find this gene in, right, we would have this double-stranded, double helix shape that DNA is. Right? This is the shape of DNA. It's this crisscrossy, double-stranded thing. And if we were to zoom out even further, right, and we're looking at this big ball of DNA as a whole, right, so this double-stranded thing kind of intertwines with itself, wraps around itself, gets tight in some places, gets loose in others. And that collective whole ball of DNA, that double-strand ball, is our chromosome. And so on one chromosome, there are many genes, because right, it's a very long, double-stranded thing with lots and lots of genes and hundreds of thousands of nucleotides, right, for gening. Then we are asked to compare DNA and RNA, their structures and their functions. The primary difference between them is that DNA is double-stranded and RNA is single-stranded. And each of these setups, these structures, have its benefits, right? And so the function of DNA is to hold our genetic code for a long time, for the entirety of our life, right? We want to keep sure that code safe, make sure it doesn't get altered by anything, it's not getting ruined, it's not getting broken. And having that double strand is nice and solid. It keeps all the goodies safe, right? All the goodies is tucked on the inside of that strand, so it's cool. This is good. The RNA's job is to serve as a messenger of sorts. It brings the information from the DNA out into the cellular areas where it can actually be utilized. And so our DNA holds all of our instructions at once, and our RNA is just the instructions for a very specific protein, controlling a very specific trait. That's our mRNA. When we talk about RNA in general, we're usually talking about mRNA. There are other RNAs. The tRNA is another one that we need to know for the story of transcription and translation. But there are other RNAs as well. And so here you see I listed the function as being short-term transfer, which is what we usually think of. But there's also modification roles that RNA play in the expressions of genes. And so some RNAs block DNA transcription. Right? Some RNAs will glob onto mRNAs and slow them down so they don't get expressed as quickly. So there's a lot of different things that RNAs are doing. But for the sake of this unit, for the sake of this course, you just need to know that it's a short-term messenger type of molecule. The RNA is not long-lived. It's not particularly stable. Right? All of its informations are just kind of naked and out there. So this mRNA will fall apart fairly quickly compared to DNA. Okay? Short-term messenger. Our next prompt asks us to model transcription and translation. That is protein synthesis. Right? The whole purpose of having this genetic code is to hold the instructions for building proteins. All of our trait expression is done via protein. So any trait you have it because the protein that is associated with that trait has a specific shape. The instruction for what shape that protein is going to be is the genetic code, which moves from DNA to RNA. The RNA is used to build the protein. So the first step in this whole process, taking DNA, reading it, using that information to build RNA, is called transcription. And it's achieved primarily by an enzyme called RNA polymerase. The RNA polymerase goes in, opens up the DNA strand at a specific gene that I want to turn on, and it reads one side of the DNA. There's a coding side, and it reads it. So when this RNA polymerase opens this up, it will read A, and it will collect a U from the environment around it. It moves down, and it will read a T, and it will collect an A. Moves down, reads an A, will collect a U, so forth and so on, okay, until it's built this whole mRNA. That mRNA then pops out, the DNA closes back up, goes back to normal, and this mRNA leaves the nucleus. It's now holding the instructions for that one specific gene. And so it took the information from the DNA, copied it over, and now it's taking it out into the cell's cytoplasm, where it's going to meet up with a ribosome, which is a type of organelle, very primitive organelle. Prokaryotes have ribosomes. One of the first organelles that would have ever existed would have been a ribosome. This ribosome latches on to the mRNA, and it starts reading its code. And so it reads the code in groups of 3. These groups of 3 are called codons. So it takes 3 nucleotides to make one codon, and one codon codes for one amino acid. You put together a bunch of amino acids, and you get yourself a protein. So the order in which the RNA nucleotides show up tells the ribosome which amino acids to put in what order. The order of amino acids determines the shape of the protein, and the shape of the protein determines its function. So you'll see here in this model, this is the mRNA. It's going through the ribosome. The ribosome is reading the codons, utilizing tRNA and anticodons, which are associated with specific amino acids. The amino acids are being linked together by the ribosome, and they come out this one gap. And so it's building this long chain of amino acids. Once the amino acid is long enough, or once the amino acid chain is long enough, it begins folding up into a ball. And that ball has a specific shape, and that specific shape gives it a specific function. And so once it gets that shape, it is the protein. Right? Prior to getting the shape, it's a polypeptide. Once it has its right shape, it's now the protein, the end game of this whole thing. That does stuff. All right. Our DNA, which controls this whole story by being the initial code for all of our traits, can be altered and changed. And when our DNA is changed, it impacts the shape of the protein that is made at the end, and by changing that shape, we change its function. There are 3 types of what we call point mutations, And point mutations are when you substitute or impact or change one nucleotide within the DNA. Okay? And so if I had an A here and I change that A, that is a point mutation. We also have chromosomal mutations, which change the whole chromosome, but that's not really tied into translation and transcription because it's too big of a change. Right? These changes are smaller in scale and have direct impact on this whole process of protein synthesis. Now, the first type of point mutation is called an insertion, and that's when we take a nucleotide from the outside and we plug it into the DNA sequence. Right? Just plug in one that doesn't belong there before. It stretches out that sequence right there. Okay. The second is called deletion, which we take a nucleotide that's inside of the sequence and we remove it. Boop, bye. And the last is called substitution, where we take one of the nucleotides in the sequence, pretend it's an A, and we swap it out for another one. So this was an A, it's now a T. Substitution. Substitutions have the potential to be completely 100% harmless. Sometimes, when you make that substitution, it doesn't change the amino acid that is being coded for at all. And we call that a silent mutation because you can't see its outcome in the protein. The protein looks exactly the same. Deletion and insertion, however, are almost always detrimental to the shape of the protein. Because when you take one out or you put one in, you shift the whole code down by one nucleotide. And so when we looked at the codons, they were in groups of 3, right? That little grouping right there. Okay? It all gets shifted down one. Right? And so now my group of 3 is off by one nucleotide. Right? And so now it used to be these 3, but then now it's these 3. And that happens all the way down the entire strand of this gene. And so it messes up all of the codons all the way down. Big deal. Protein's going to take on a whole new shape because of it. We call this frame shift, that shifting of codon groupings. It's called frame shift. And it's almost always a bad thing. Okay? The last point for this unit is just asking us to explain how epigenetic traits work. Epigenetic traits are traits that are impacted by the environment. Right? So the actual phenotypic expression of a trait is altered based not upon a change in DNA, but by an environmental factor. A really easy example of this is tanning. Right? And so our skin tone is a phenotypic expression of a specific trait, of our melanin expression. Right? When I go out in the sun and I get the beautiful bronze that obviously I do, it's not like my DNA has been altered to make me more tan. What has happened is the expression rate of that DNA has been upped. Right? So my phenotype changes, but my DNA has stayed the same. So that's a really simple example of epigenetic traits. There are some much more complex mechanisms that are available for epigenetic regulation, but honestly, that's a little bit beyond the scope of this course. That's another thing that if you really want to find out about, you're more than welcome to take AP Biology your senior year. I'll be happy to have you. I'll be happy to teach you all about epigenetic traits. I love epigenetic traits. I think they're fascinating. But yeah, so that's it for Unit 7. We done. Bye.